Assalamualaikum Dr. AZIYAH. My name is Nushara binti Zahani. Today my group and I will be present about the article Clonal spreading of ST42 Staphylococcus haemolyticus strains occur possible due to fusB and tetK resistant genes and capsule-related genes. This is the overview of the presentation. This study was initiated based on the hypothesis that the presence of fusB acid and tetracycline resistant and capsule synthesis genes in ST42 Staphylococcus haemolyticus strains may lead to the prevalence of the strains.
The objective of this research are, firstly, to analyze the different molecular types of Staphylococcus haemolyticus gene structure. Secondly, to understand the distribution of antibiotic-resistant genotype and phenotype among collected ST3 and ST42 clinical isolate. Thirdly, to identify the specific antibiotic-resistant gene and virulence that may be responsible for ST42 prevalence.
This is the article's list of procedures, but only 3 significant methods will be explained. First, whole genome sequencing and annotation. This method aims to examine a cell genome DNA sequence and identify functional elements along the genome sequence. Using the PET-BioTM method, the complete genome sequences of S. haemolyticus strains were analyzed. The raster annotation service was utilized to validate the annotated data and determine the function of each gene. Second, the antimicrobial stability assay to observe the sensitivity of strains to antibiotics containing fusaric acid, clindamycin, erythromycin, and tetracycline. The discs were placed on the top of the bacterial growth media. The sensitivity of the bacteria towards the antibiotic was evaluated using the inhibition zone. Last is polymerase chain reaction. To amplify a DNA sequence and characterize of the isolate strains drug-resistant genotype and virulence factors. The PCR detects drug-resistant genes including MPHC, MSRA, and CAT. Specialized PCR priming sets were utilized to detect the AD1-LP fusB fragment, which encodes for various types of fusaric acid-resistant isolate.
There are 4 major results that has been revealed in this study. Firstly, genomic similarities. The strain of ST3 and ST42 show the similarity in terms of general genomic characteristics, such as the number of coding sequence, tmRNA, tRNA, rRNA, and GC content. They show these strains have a common genetic backbone. Secondly, fusaric acid resistant. The study found that fusB staphylococci was more prevalent in ST42 compared to ST3. This indicates that ST42 strains may take advantage of fusaric acid resistant for clonal spreading. Thirdly, tetracycline resistant. The distribution of the tetK gene and the tetracycline resistant phenotype was much greater in ST42 compared to ST3. From this result, we may know the genetic characteristic and antibiotic resistant profile in different lineage. Lastly, capsule formation-related gene. The 4 genes which are involved in capsule formation and bacterial adhesion were found to be more prevalent in ST42 compared to ST3. For the interpretation of result, the combination of multiple drug-resistant gene, the presence of capsule-related gene, and the virulence factor in ST42 of Staphylococcus haemolyticus strains likely contribute to their successful clonal spreading in Taiwan. This factor enables ST42 to survive in challenging environments, including exposure to antibiotics and interaction with the host immune system. The result can be interpreted differently. First is alternative mechanism of resistance. There are other resistant mechanisms that exist beside fusB and tetK. This is because there are mutation and extra resistant genes based on the observation drug-resistant phenotype. Hence, it is crucial to analyze genomic and functional characterization. Second is complexity of virulence factor. The functional significance and contribution of these factors to the overall virulent ST42 strains require further investigation. Animal model study and activity of this virulent factor would provide a more comprehensive understanding of their role in pathogenesis.
Hello, I'm Ain, and now I would like to present some key points regarding the discoveries on S. haemolyticus strains. So the first point we'll discuss is the evolution of S. haemolyticus strains. Over time, these strains have evolved from the original ST3 strain to various molecular types, with ST42 becoming the predominant lineage in Taiwan. Moving on, we'll talk about the clonal spreading and also drought resistant. It has been observed that ST42 strain, in comparison to ST3, possesses a higher number of drug-resistant genes. This suggests that ST42 may have an advantage in competing with other strains. So plasmid structure comparisons have also indicated that the involvement of the mobile genetic element, MGEs, in transferring antibiotic-resistant genes. Now, let's focus on the fusidic acid resistance, an antibiotic commonly used to treat staphylococcal infections. Interestingly, ST42 strains exhibit a higher rate of resistance to the fusidic acid. This resistance is facilitated by the presence of the fusB gene. It is believed that this resistance contributes to the clonal spreading of the ST42 strains. Lastly, we'll discuss on the role of capsule-related genes and virulence factor. So most ST42 isolates carry genes associated with capsule synthesis and a specific virulence factor called CLFP. These factors may contribute to the increased virulence and also the ability of the ST42 strains to cause infections. There are external sources providing strong evidence supporting the virulence of ST42 strains, such as comprehensive studies in China that found cap genes in ST42, and also an animal model showing higher virulence compared to non-ST42 isolates. In this study, the collection of the isolates was limited to 2 medical centers in Taiwan, which means there is a lack of diversity in representing the S. haemolyticus strains from different regions. So to gain more comprehensive understanding of the strain diversity, it will be beneficial to include isolates from more medical centers. Next, the relationship between the genome sequence of SH51 and SH29 is uncertain. So previous studies did not mention a similar plasmid in SH29. So further analysis is necessary to determine the origin and also the relationship of PCGM SH51 with SH29 and also CGM SH51. Next, the study also lacks an animal model to study on how the 3 capsule synthesis related genes contribute to strain virulence.
The conclusion of this research are the research revealed 2 drug-resistant genes, fusB and tetK, that has higher preference rate in ST42 compared to ST3. This the research suggested for clinical treatment combination with multi-antimicrobial drugs. In my opinion, I agree with the conclusion from this research. The high rate preference of both drug-resistant genes in ST42 can lead the strain to become the antibiotic-resistant gene. With the detected of a capsule synthesis-related genes and adhesion factor that protected the ST42 make the strain become more resistant to antibiotic.